Okay, so we've been talking a whole lot about the relationship between genes and proteins and how they code for traits and how you are a combination of two parents, their traits combine to form you. But what we really want to talk about is like, okay, how does that actually play out in like looking at the big picture? Like, why do I have my mom's eyes but not my mom's nose? Like, how does that all work? So what that's called is heredity. And again, this is all review. This is all stuff we've seen. So if I go through it a little bit too quickly, reach out to me and I can re-explain it, and we're going to practice it tomorrow, but this is all review. So we've talked about heredity, which is again just the passing down of the traits from a parent to an offspring. When we first learned about heredity, we learned about this man with an ugly name, Gregor Mendel, also kind of an ugly face, no shade. But he is the scientist who, a long time ago, did a lot of experiments and discovered all of what we know today about heredity. And he did this by studying and doing experiments with these different pea plants. So he used these pea plants and made them reproduce with each other and make more and more pea plants. And he noticed that there seems to be different versions of each trait. So for example, if we look at like the pod color of a pea pod, he noticed, okay, sometimes they're green and sometimes they're yellow, or the shape of them, sometimes they're inflated or sometimes they're constricted. So he figured, after all of this studying, after all these experiments, he figured that there must be two different factors that determine each trait, and you probably get one from each parent, right? So he was totally correct in this. He knew what he was talking about. Those factors that he's talking about that you get from each parent, we know those are your genes, right? So you get some genes from your mother and the rest of your genes from your father, 50-50. And the different versions that those genes can be, that's what we call those alleles, which I know is a word that we struggle with, right? So an allele, like I said, it's just a different version of a gene. So you can have a gene for eye color. So the different versions of eye color, we know this, you can have brown eyes, you can have blue eyes, green eyes, gray eyes, those are all different versions of the same gene. Same thing if you look over here with hair color. The gene is for hair color, but the different versions or alleles could be blonde, red, black, brown, all of these different versions or alleles. So like I said, you get 50% of your alleles from your mother, 50% from your father. So they combine after that sexual reproduction happens to make what we call your genotype. So your genotype, again, is just the combination of alleles or the different versions of traits that you inherit from both of your parents. So once you have your genotype set in your chromosomes, they are going to do their job, code for those proteins which determine your traits, right? So once your genotype is set, it's going to determine the traits that we actually see, which is the phenotype. So if you have a genotype to code for brown hair or brown eyes, the phenotype is when you're actually going to see, oh, this person has brown eyes. You don't see what's actually written in the instructions in the gene, right? That's kind of weird. What we see, the actual physical traits that we can observe, those are our phenotypes. Now, like I said, we get two alleles from each parent. So you get one allele from your mother, one allele from your father. They can be either dominant or recessive. We remember the dominant allele we represent with a capital letter, and the recessive allele we represent with a lowercase letter. So as long as you have just one dominant allele in the genotype, you are going to see that dominant version in the phenotype. So what do I mean by that? If you look here at this picture of these roses, so we have the dominant allele, which is a capital R, and the recessive allele, which is a lowercase r. So the dominant allele in this case is going to be the red color rose, right? So here, this flower inherited two dominant alleles, so it's going to have a red rose. But over here, these roses only have one dominant allele. They also have a recessive allele, but it doesn't matter because as long as you have just that one dominant allele, it's going to, like it sounds, dominate over that recessive allele. So that color is going to show up. So even though it has only one dominant allele, you're still going to see that red color. The only time you're not going to see the dominant allele is if there are no dominant ones present in the genotype whatsoever. So here we have two lowercase r's, right? So that's two lowercase or two recessive alleles. So you're going to see the recessive phenotype, which in this case is a white flower. So again, you're only going to see the recessive allele in the actual phenotype if you have two copies. There cannot be any dominant alleles or it will dominate. So we also have a few fancier terms to describe the different types of genotypes you can have or the different combinations of alleles you're going to get from your parents. So if you have a genotype with two copies of the same allele, meaning they're either both uppercase or they're both lowercase, we call that homozygous, which we talk about it all the time. The prefix homo just means same, so when you see homozygous, think same exact alleles. So in this case, we have two uppercase or dominant, so we would call this a homozygous dominant genotype. Or over here, we have two lowercase or two recessive, so this is homozygous recessive. Now, if they have one of each, so one dominant, one recessive, that's what we call heterozygous. Because hetero, that prefix hetero means different, right? So we're going to have two different alleles, so a dominant and a recessive. And again, since these have two dominant alleles, this will show the dominant phenotype. Over here, the heterozygous will also always show the dominant phenotype because it has a capital letter in it. The two lowercase a's, however, the homozygous recessive, will always show the recessive phenotype because it has no big letters there dominating over the tiny letters. Here's your beautiful cheat sheet. I really like this one personally. So again, just a reminder, the dominant allele is the big letter, recessive is the small letter. Anytime there's just one dominant allele around, one big letter, it's going to literally dominate or take control over the smaller letter. So think of it as like the capital A or the dominant allele is like a really big, strong person, and it can easily beat up this recessive allele. Like this recessive allele will never win against the dominant allele, right? So the recessive can only take over the phenotype if there are no big letters around. So they can kind of be hidden, which we're going to talk about in a second. To remember the different types of genotypes, again, just remember homo means same, hetero means different, and heterozygous, this is what I want to talk about. You might see heterozygous called a carrier. That's because they literally carry around a recessive allele with them. So while the genotype for a heterozygous will show that dominant trait, like in this case, if the dominant trait is brown eyes, it's still a carrier for this recessive allele. So it's carrying around a recessive allele for maybe blue eyes, but we just don't see that. So heterozygous you might often hear called a carrier because, again, they're carrying around that little letter. Okay, now here's the thing that some of us like, some of us don't like. It is what it is. Punnett squares. Why are Punnett squares even important? Why do we use them? So, I can't even spell squares. That's embarrassing. Okay, so Punnett squares. These are just charts that we will use to predict what a possible offspring will get from each parent. So if we know the genotypes of both of the parents, we can kind of guess what different combinations of alleles that offspring will inherit from both of the parents, right? So again, Punnett squares show all the possible allele combinations of the offspring because you have one being donated from one parent and another allele being donated from another, and it can help you find the probability that a certain genotype will occur, which probability, if you remember from math class, sorry to bring more math into this, but it can be represented in either a fraction or percentage, and we'll practice both, right? So how do we even use a Punnett square? What the heck? We put the genotype of one parent at the top outside of the box and the genotype of the second parent down the side. And inside of the box represents all of the possible genotypes of the offspring. So outside of the box, we're talking about parents. Inside of the box, we're talking about the kids, the offspring. So we remember when we're setting up our Punnett square, though, we separate the alleles in the genotype. So let's just do this example. If we have one parent having a homozygous recessive genotype, meaning 2 lowercase b's, we'll put the first allele on top of the first box, and then the second allele, which is another lowercase b, in the second box. Okay, so never put both letters above one box. Incorrect. Parent 2, we're going to do the same thing over here. The first letter is a capital B, and the second letter is a lowercase b. There we go. So then to fill out your Punnett square, you literally just look up and to the left to figure out what letters we are typing. So in this first box, if we look directly to the left, we've got a big B, and above it here, we've got a lowercase b. Same thing for over here. Look all the way to the left, we've got a big B. Up top, we've got a lowercase b. And then same thing down here, look up and to the left, so we've got 2 lowercase b's, and to the left and up, we've got 2 lowercase b's. So we've got 2 heterozygous and 2 homozygous recessive. So then these, again, are the possible genotypes the offspring of these 2 parents could have. So this is what they could potentially be carrying in their genes. So the question actually asks, what is the probability that the offspring of 2 parents with these genotypes, what is the chance that they will have brown eyes? So we see here that the dominant allele is brown and the recessive allele is blue eyes. So again, as long as there's just one dominant allele present in the genotype, you're going to see the dominant phenotype, which is brown eyes. So here we have 1 with the dominant allele and 2 with the dominant allele. So you always want to count how many out of 4, because there's 4 chances, right? So we have 2 out of 4, which is the same as 1/2. And we know 1/2 as a percentage is 50%. So there's a 50% chance that the offspring of these 2 parents are going to have brown eyes, which means there's a 50% chance they'll have blue eyes. Kind of cool. So that is the whole point of the Punnett squares. Here's your cheat sheet. It's going to help remind you how to actually set up a Punnett square. Here's where the genotype of parent 1 and parent 2 goes, and these are the possible genotypes of the offspring. When do I even know to make a Punnett square? So if you ever see these phrases in any type of test question, you should know, I should get out a piece of paper, I'm going to have to make a Punnett square, right? So it asks, what's the genotypes of the offspring? How many of the offspring would have this trait? What are the chances of the offspring having? If you see anything about chance or genotypes of an offspring, we know we're going to have to make a Punnett square, right? And this over here, again, just shows how to answer different questions about Punnett squares. So really, I know I gave you a lot of information today, but we're going to practice it tomorrow. I think Punnett squares are things that get easier with practice, so I'm not worried about us. I think we got this down. Hit me up if you have any questions.